Good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Expansion Cohorts in Early Development of Anti-Cancer Agents. My name is Edward and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive and webinars work best when you're involved, so please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in a listen-only mode. Please note that this event will be recorded and made available for future streaming. A big thanks to IDDI who developed the content for today's presentation. The International Drug Development Institute is an expert center in biostatistical and integrated e-clinical services for pharmaceutical and biotechnology companies in several disease areas, including oncology and ophthalmology. IDDI optimizes the clinical development of drugs, biologics, and devices thanks to proven statistical expertise and operational excellence. Founded in 1991, IDDI has offices in Belgium, Boston, Massachusetts, Raleigh, North Carolina, and San Francisco, California. And now I'd like to introduce our speakers for today's event. Everardo Sad has over 15 years of experience in medical oncology and clinical trial designs. He graduated in medicine and trained in internal medicine in Sao Paulo, and also did his fellowship in medical oncology at the University of Texas Anderson Cancer Center in Houston. After practicing for several years, he shifted his professional career towards education and research in medical oncology and has a special interest in clinical trial methodology, the assessment of endpoints, and the development of novel therapies for cancer patients. Elizabeth Cohort is a consultant in the design of early to late stage clinical trials in various therapeutic areas. She brings over 10 years of experience as a statistician for biotech and diagnostic industries with a special interest in biomarker validation and biomarker-based clinical trials. Els also has a strong background in applied biology and assay development. And now, without further ado, it is my pleasure to hand the mic over to Everardo Sad, who will be starting off today's presentation.
Thank you, Edward. Hello to all of you. Thank you for attending this webinar. So my task is to provide you with an introduction where I will basically show a few recent trends in early development in oncology, particularly with regards to expansion cohorts within phase 1 trials. And then I'll just briefly highlight the main points of the FDA guidance on this topic. When I hand it over to Els, the plan is for her to make several considerations regarding sample sizes and the desirability of having randomization in these studies. And then she'll briefly touch on additional statistical issues. So just by way of introduction, phase 1 trials traditionally have been around for a long time. And of course, not all of them are first in human studies. Some of them are on still novel agents, but not necessarily the very first trial. Some of them are for combinations, sometimes involving not only systemic agents, but radiation therapy, for example, and sometimes for special populations such as children or people with organ dysfunction, for example. And then traditionally, there has been something called phase 1/2 trials, which were more like phase 2 studies in which there was a need to start and perhaps confirm safety in something that looked more like a phase 1 study, but then quickly we would move on to a phase 2 assessment when this was considered safe. And then more recently, phase 1 trials with expansion cohorts came along, and I think they sit sort of between these two trial types. And just to be clear, this is what we mean by expansion cohorts. So you have a phase 1 study with a dose escalation phase, for example, represented here by progressively darker shades of blue. And once the recommended phase 2 dose is achieved, you start cohorts of different disease types, for example. Not all of them may reach the end of the planned accrual, depending on the statistical design, but this is what we mean by expansion cohorts. And in fact, this type of trial has been around for some time. They're not so new. So in this quite well-known paper published more than 5 years ago, the authors have looked at all published phase 1 studies and then tried to see the prevalence of phase 1 studies with expansion cohorts. And as you can see, as early as 2006, there was already 12% of phase 1 trials with this feature. But it's true that the prevalence of expansion cohorts is growing along the years, so they're becoming more frequent in oncology. And there are some features that are more characteristic of trials with expansion cohorts. For example, they are more frequently multi-center, as shown here, sponsored by pharmaceutical companies, and also they more often assess drugs which are not classic cytotoxics. So in other words, non-chemotherapy type of agents, such as targeted agents or even immunotherapy. Now, expansion cohorts is somehow associated with increased success. Of course, we can never tell whether this is a causal association, but it's true that trials with expansion cohorts have been more successful. So these authors have looked at the fate of drugs that were assessed in phase 1 trials with expansion cohorts. So in comparison with drugs that did not have an expansion cohort, those with these cohorts had higher response rates in phase 1 and also higher success rates in phase 2 and even approval rates by the FDA. So for example, here out of 381 drugs, out of 112 drugs that were subject to a study with an expansion cohort, 21 were approved versus only 13 drugs out of 269 without an expansion cohort. So there is clearly an association with success in expansion cohorts. Of course, this may be confounded by several factors, especially the type of drug and the intent to conduct an expansion cohort study. We all know that the face of oncology trials is really changing, and it's changing relatively quickly. And this paper that was written by the FDA some years ago nicely summarizes the main changes. Two of them are shown here. One of them is the progressive loss of demarcation we can see here. Traditionally, phase 1, 2, and 3 were more separated, whereas now there is more of a seamless transition between phases. And the other major change is the fact that efficacy has become really of more concern in phase 1. Efficacy has typically been a phase 2 endpoint, and of course phase 3, but more recently, the assessment of activity has become an important component, let's say, of phase 1 trials. Now, this has led to the famous pembrolizumab phase 1 study. We cannot talk about this subject without mentioning this trial, because it was the first in-man study of this checkpoint inhibitor, and it started out as a study that planned to enroll 30 patients. And through several amendments, it ended up with more than 1,200 patients. So clearly this is an exception and not the rule, but it was a trial that really made the headlines in several ways because the drug was very, very active. And this was really the driving motive for expanding this study so much through several cohorts, and even some of them randomized between different doses of the agent, not against a control arm, but different doses of pembrolizumab. Now, in part, I guess, because of situations like this, the FDA started to express some concern, or at least feel the need to provide some guidance for sponsors who wanted to conduct phase 1 trials with expansion cohorts. And as most of you will know, this guidance came up late last year, I believe, and from now on, I'm going to make a few comments about some of the highlights of this document, which is, by the way, a very well written document that addresses essentially only first-in-human studies. Apparently, the concern is really with first-in-human trials, so not all phase 1 studies, not all phase 1 trials with expansion cohorts. And the goal of these trials in the FDA's view is really to expedite drug development by seamlessly proceeding to a situation where you have a trial that is more of a phase 2 type, and so these are the expansion cohorts. Now, in this document, it's very clear that the major concern, the major considerations are about safety, and also about the logistics of conducting these trials, because the expansion is done at a time when not a lot is known about the new drug. So there are concerns about safety, of course, and also about the difficulty that investigators may have in disseminating information to all the participating centers and physicians in a timely manner. So a lot of the considerations on this document are about safety, about the challenges and risks of exposing patients to suboptimal on the one hand, or toxic on the other hand doses, exposing perhaps more patients than required to achieve the cohort's objective, the possibility of misinterpretation from, let's say, unplanned analysis, for example, in terms of comparing the activity of the drug in different subpopulations, whether defined by biomarker or by disease type. And so they recommend several strategies to reduce such risks, and this is a very important set of considerations for sponsors thinking about conducting a study like this. Now, we believe that the draft guidance is somewhat lean or somewhat poor in terms of statistical considerations. Although there are some, I think we think there is room for some more, let's say, at least comment, if not improvement. So they mentioned that there needs to be a statistical analysis plan that clearly justifies sample sizes and perhaps stopping rules for futility in each cohort, the magnitude of activity that would be, let's say, indicative of the need to continue to develop the drug versus not continue to develop. They caution against comparing between cohorts if there has been no random assignment. And the only part where randomization is really mentioned is between those levels, for example, of the agent. And so there is no mention of randomization to a control arm. Now, one may wonder if it makes sense to randomize in phase 1 a situation where at least traditionally patients have exhausted their options in terms of standards of care. But it's true that oncology is changing in that regard, and very often nowadays patients in phase 1 studies are in earlier lines of treatment than they were before, just because new drugs are being developed that seem to be at least more promising, or that have very specific targeted actions. And so the selection of patients has changed, And it's true that there may be several situations where patients have not exhausted all their standard possibilities. So having all these considerations, IDDI wrote a critique or a comment to the guidance, and this is available online in the link that is provided on this slide. But essentially, we comment and propose the possibility of increasing the use of randomization in expansion cohort studies. So before I hand it over to Els, this is sort of a situation that may be confronted by some sponsors. So you have a potential dilemma between at least two situations. So you have a new drug, and then you are encouraged to conduct an expansion cohort study. And so you wonder, should I randomize or not? So on the top panel, we see a more typical situation with no randomization, perhaps with stopping rules that led to closure of the disease C arm. But you have patients treated with the experimental agent in the 3 cohorts without randomization. You may have possibly fewer patients by doing that. It might be easier to enroll, arguably, in some settings. And so you might think this is more efficient. On the other end of the spectrum, you may have a properly randomized trial, again, with perhaps stopping rules for futility, which will allow you to have a better estimate of the treatment effect, as Els will show us. So this might be possibly more informative. So the question is, what should a sponsor do in this case? And now I'll hand it over to Els, who will address the statistical, let's say, issues around these studies.
Okay, thank you, Everardo, for this nice introduction. So indeed, I will look into more into detail in the statistical properties of this expansion cohorts and a randomized clinical trial. So from a statistical point of view, an expansion cohort, it's nothing else than a single-arm design that we all know. And in oncology, they have been around for a long time, and they are typically implemented with as primary endpoint the overall response rate. Now, the attraction of this expansion cohort is mainly in their small sample size that might be seen as more efficient. And this is because what you do is you compare your experimental drug to an historical control that is not internal to the trial, but I will come to that. In the FDA guidance, they propose the Simon two-stage design as a good design for an expansion cohort, and this has been criticized in some of the comments that were sent to FDA on this guidance, and it was felt that it was a bit too restrictive because it's a well-known, it's a proper design, but there are other designs and science is evolving, so it might be too restrictive to only in the guidance document refer to this one classical single-arm design. Now, what Simon's two-stage design has in common with many other single-arm designs is a particular way of specifying the null and the alternative hypothesis, which is not the same as in a randomized clinical trial. So here you see that the null hypothesis is specified in terms of a response, a proportion that does not warrant further investigation, the P0, and the alternative hypothesis is specified in terms of another proportion, another response that warrants further investigation. Now you see that there's a range of responses that is not included in those hypotheses, and this means that we have to be careful with how the results are interpreted. The correct interpretation when you reject a null hypothesis is that you have shown that the response rate is higher than this uninteresting response rate, and that the data are not inconsistent with your anticipated promising response rate. But this does not mean that you have shown that that you can accept your alternative hypothesis and that you can say that you've proven that your response rate is above this interesting this interesting proportion PA. So it is very crucial for this type of designs to carefully decide where you want to put the bar for these P0 and PA, because otherwise the trial might not have much purpose. So often this P0, this uninteresting response rate, is chosen close to the response rate that will be achievable under the standard of care. And if you can reject this response rate, then this could trigger further studies, further investigation of the drug. But I will come back to this. It does not mean that you have strong evidence that the next trial will be positive. Now, throughout the webinar, I will, to address the dilemma that Everardo introduced, I will use the same case study. And it's a case study I think that will be familiar to many statisticians and other professionals in the clinical trial field. It's a sponsor who wants to develop a drug that when it is given together with a checkpoint inhibitor, is assumed to increase the response rate in metastatic non-small cell lung cancer patients that have a PD-L1 positive marker. Now, we assume that the dose has been selected in the first part of the study, and the current standard of care for those patients is the monotherapy of the checkpoint inhibitor. Based on historical data, the sponsor estimates that the response rate under standard of care will be around 40%, and they consider an absolute increase with 20% as a clinically meaningful increase. Now, if we would develop, or if we would design a Simon's two-stage trial for this setting, then it could look like this. So you see here on top that the type 1 error is set to a very liberal one-sided 10%, reflecting the early stage of the development. Of course, this is something that the sponsor can decide upon. And you see in the definition of the null hypothesis, you see this 40% or historical control is mentioned. And in the alternative hypothesis, we mention the response rate that we assume to be clinically meaningful. The Simon two-stage design is, of course, a design that goes in 2 stages. First, we enroll patients that are all treated with the combination of the checkpoint inhibitor and the experimental treatment, and the trial continues until 12 patients have been included and treated. And here there's a futility analysis. If the number of responses is too low, so lower than 6 patients show a response, then the trial is halted for futility. If we have 6 or more responses, the trial continues to enroll patients for a total of 38. And then again, based on the number of patients that reach a response, the trial is positive or negative. Positive meaning at least 19 responses, and that would warrant further development of the drug. Now you see that the total trial only has 38 patients, and also, like I mentioned before, it would be called positive, or it might be indicative of warranting further development if you observe a response rate of 50%. So at this point, and I see my confidence interval dropped off the slide. At this point, the confidence interval for this response rate would exclude the historical estimate of 40% under standard of care, but it's still lower than our design proportion in the alternative hypothesis, which was 60%. So our data are not incompatible with this response of 60%, but we did not show that this response rate is 60% or higher. Now, you see that the setting of this null hypothesis, where we would set the bar, is very important because it decides on the outcome of the trial. And here, in this case, the sponsor, they looked at previous clinical trials with this checkpoint inhibitor, and looked at patients with different levels of the biomarker and looked at a number of responses. Now, you see that the number of patients that were treated, they were not too high. And so when they picked the historical control rate and fixed it at 40%, you can see that it's really, they picked it somewhere in the middle, but there was a high variability or a high uncertainty around it. The other aspect with this historical control is that although they used clinical trial data as the historical control, maybe these trials, they had slightly different inclusion-exclusion criteria. So maybe they are not sure that these specific subsets of patients they are interested in, whether these are the same as the ones that were treated in the previous trials. So because of all this uncertainty, the sponsor might be interested to see how many patients one would need for a randomized comparison study. And here I designed the study as a classical randomized comparison study. I used the same liberal operating characteristics. And you see here that although I still assumed the same performance under the different treatments, we no longer see this coming back in the definition of the null hypothesis or the alternative hypothesis. And that's because for a randomized comparison, it's about the difference between the arms, and it's not about one arm reaching a certain number or a certain proportion of responses. So I included here also a futility just to compare with the Simon two-stage. I'm not saying this Brian-Fleming boundary is the best, it's just as an example. And here, so the patients would be randomized. I use the 1-to-1 ratio here, which of course can also be altered. Half of the patients in this case would go to the standard of care, the monotherapy of the checkpoint inhibitor. The other half would go to the combination with a new agent. The interim analysis is after enrollment of the 58 patients, and here it's not about responses but about the difference. If there's a difference of 6% in the responses observed at this point, the trial continues. If the difference is less, then it stops. If it continues, you would need a total of 116 patients, and then in the end, again, the difference is compared, the difference is evaluated. If it's 12% or more, this would mean that you have shown a significant difference in response between the arms, and the drug would warrant further development. Now, of course, there's a huge difference between the 116 patients needed for the randomized comparison and the 38 patients that were needed in the single-arm design. But this is because here the variability of the control arm comes into play. We do no longer assume that is in the control arm the response rate is fixed and it's 40%. No, we will estimate the response rate in the control and compare it to the response rate in the experimental arm. So here success is about differences in response rate between the arms. Now, the sponsor, as many sponsors that we interact with would do, in this hypothetical case, they selected the expansion cohort with the Simon two-stage design, but they said only 38 patients, so there's room for another cohort, and they decided to simultaneously run another cohort, this time in the PD-L1 negative patients, for which there's another standard of care. It's then the CPI plus the chemotherapy, and they would add their agent to this. Now, following the suggestion of the FDA guidance document, they conceived the trial as 2 independent single-arm trials, and they implemented both with the Simon two-stage design, which would bring the total sample size of the trial to 72. And this is because the historical data also showed that for this subgroup, the response rate would be around 40% under the standard of care. So now we have a study with not only one, but with multiple cohorts. And this is again nothing new. For example, we have recently heard a lot about basket trials. That is one form of a design with different cohorts. And it can indeed be constructed as a series of independent phase 2 trials within each of the cohorts. But there are many alternatives that will allow you to borrow information across the cohorts. So this borrowing can be formal borrowing of information, and then we are in the Bayesian scene, and we look at the Bayesian designs. I mentioned here just one example. There are many more. The Bayesian basket trial, this is also a Simon design, but now the Bayesian version. Another way of borrowing information across cohorts can be informally. And it can be done, for instance, by applying a Sargent's three-outcome design to each of the cohorts. Here you add a third possible outcome for the study that is inconclusive. And if one of the cohorts falls in this inconclusive zone, you can informally borrow information or look at the results of the other cohorts to make your decision about the development of the drug for that particular cohort. Now, of course, especially in this formally, in these Bayesian designs and other designs that formally borrow information across cohorts, these type of designs, they can become complex, which is not a problem, but we should always be aware that there's a trade-off between the efficiency they might bring and the operational complexity, and maybe also the complexity in communicating the results and so forth. So we prefer to keep our trials simple if possible, but only make them more complex if it brings sufficient efficiency. Now, back to the case study. So the sponsor decided to go for these two single-arm designs with 72 patients. And in this case, because we feel strongly about the potential problems and related to the uncertainty of this external control that was used to set the null hypothesis, we at IDDI often propose an intermediate solution between this expansion cohort or single-arm design and the fully randomized comparative study. This intermediate solution goes as follows. So we don't redo the sample size, we keep one single-arm design. So that would be, in this case, the Simon two-stage design. And this is just copied, so this remains the same. But we include a 2-to-1 randomization in favor of the experimental arm. And so we include this control arm with the standard of care. This control arm is not meant to be a formal comparative arm. For this, the study would not be powered, but we see it as an internal calibrator or a concurrent control, and that will help to evaluate the reliability of the historical data. So in fact, the reliability of the design and the null hypothesis we took for this experimental arm. Let me now look at how this internal calibrator arm can be informative, how it could be more or less efficient if we compare it with the expansion cohorts or the single arm designs. Now here, of note, I want to mention that the FDA guidance, they refer to outstanding efficacy results or outstanding drugs to be the most suited for this expansion cohorts. Of course, this is not according to my logic, because before the expansion cohort start, you can only have some idea about the performance, but you have no evidence. And also this, let's not forget that these outstanding results, they do occur, and that's good, but these outcomes are rather rare. So I will look at outstanding study results for our case study, but also at the common disappointing and promising results. So the trial we designed with this we think or we hope that our experimental arm will show a 60% response rate, and where we think that based on our historical control, the standard of care would result in a response rate of 40%. Now, if we observe 79%, almost 80% of patients that have a response, these results might be outstanding. It could be potentially meeting the criteria for breakthrough therapy designation, and this might be indeed one of those exceptional situations. But if the trial would not have this internal control arm, then we might not be sure that these results are truly outstanding. Let me explain. So if we have the design with our internal calibrator, and if this internal calibrator, if it performs as expected, so it has a response rate really close to what we anticipated, it gives us more confidence that the design was proper, was appropriate for this patient population, that the historical data are suitable to evaluate our study results. But even more, in this case, if the results are really outstanding, often this internal calibrator that was not meant to be a comparator, it could still give you enough statistical power to do the formal comparison. And this is, of course, perfectly fine because there was randomization and these arms can be compared. So in this case, if we test the response rates and we compare them between the arms, the test would be significant. On the other hand, if this internal calibrator would also exceed expectations, so it's not only your experimental treatment that exceeds the anticipated response rate, this could go in a direction or could give you some evidence that the historical control rate might not have been suitable to evaluate your experimental treatment. And in this case, while sponsors might be at first be disappointed to see this, and they might say like, look, if we would not have included this arm, we would have had outstanding results. Now this is of course not true, and without this internal control arm, there might have been many more patients and many more studies performed with this drug before discovering that after all this drug isn't as promising as it appeared. And in this way, this design could be highly inefficient. So I will go quickly over the other potential outcomes of our case study. So if the results are disappointing, we have only 45% of patients with a response, and the historical control says the standard of care would be already 40%. In this case, without an internal control in your design, the sponsor might decide to halt further drug development. If here, like shown here on the left-hand side, if the control arm is also in line with expectation, it means the historical data might be suitable, might after all be suitable to evaluate this study results, and it further it gives you further suggestion that it might be wise to stop the drug development. On the other hand, if your internal control is also exhibiting a lower response rate than what you anticipated, this might give you an indication that the drug after all might be active, because here we see that we have a difference of almost 20% between the two arms, which is the same as what we assumed during the design, although that for the two arms the response rates are lower. So in this case, you might still decide to further develop the drug. And for the promising results, it will be the same. So with promising results, I mean the results that I think are very positive, but will likely not be sufficient for any breakthrough designation, and for which you would need to plan additional studies. So here again, if you have an internal control arm that is in line with expectation, you can be more confident to proceed with the drug development. And here, if the control arm also has a much higher or shows a much higher response rate than what you anticipated up front, this might be a sign that the drug is not as active as you might think. And without this arm, you might set up further phase 3 trials, fully randomized, to detect the treatment effect of an absolute increase of 15%, and then this might fail. So after all these considerations and these different scenarios, I come to the closing remarks. Expansion cohorts, they are, the single arm designs. There are many different designs available, and it might be too restrictive to just look at the Simon two-stage design. There are many other flexible and efficient designs available that, of course, should then be carefully planned and designed. The attraction of the expansion cohorts lies mainly in their relatively small sample sizes, but we have to understand that it comes at the cost of increased uncertainty, and I should add a decreased amount of information, of evidence about the drug's activity. In this intermediate solution that I introduced, we used an imbalanced randomization in favor of the experimental treatment, and this, I think, has advantages in this early stage of development because it maximizes the information on efficacy and safety of the new agent, but still it has this concurrent control to help interpret the patient outcomes at the end of the trial. The randomized trials with the control arm used as internal calibrators, we feel these are an interesting intermediate solution between the expansion cohorts and the fully randomized comparative studies, regardless of the anticipated study outcome, because it helps evaluating the suitability of historical data, and it assesses the amount of selection bias that might be affecting the results in the expansion cohort. For a drug that is really outstanding, there is only a short window of opportunity for randomization after those findings. So an early inclusion of randomization might be the only chance to obtain unbiased evidence on this drug's efficacy. And if these initial outstanding results are due to selection bias or play of chance, it's better to know this upfront and not to have the need to expose more patients in more studies to discover this. In case there's no selection bias, then this intermediate type of design might have enough statistical power to do a formal comparison between the arms, and in this way provide pivotal evidence. So also for trials with other outcomes, still this internal control might be instrumental in deciding whether to stop or continue the development. So in essence, in many cases, a randomized design will be more efficient than an expansion cohort when we not only look at this one first study, this first stage of the development, but when we look at the totality of the drug development. So thank you very much. I hope we already covered some of the questions that you had, but there's now time for some Q&A, so please let us hear your questions.
All right, great. And thank you very much, Els and Everardo, for that very insightful presentation. As Els mentioned, I would like to invite our audience to continue sending in any additional questions or comments right now using the questions window for the Q&A portion of today's webinar. I've already received a few questions, and so we'll get started with those. This first audience member asking, in a non-randomized cohort, can one use real-life data as the expected outcome for historical patients?
Yes, so I think I can take this question. So, well, you can use all data from all nature to make your historical control. But of course, I think here we have to keep two things in mind. There's one, there's the precision, and the other is the bias. So with real life data, we cannot be as sure as we would be when we are using clinical trial data on how they are collected and whether they are for the population that we also target in this new design. So I think regardless of what you use as external data, it remains external data, and that's the main problem.
Great, excellent. Thank you so much for that answer. Another question here from another viewer asking, when referring to ORR, is this most often assumed to be a confirmed ORR?
Yeah, thanks for the question. Yes, in principle, yes. We're talking about confirmed responses. At least that's how they should be assessed and report. For some reason, RECIST 1.1 says that confirmation of complete and partial responses is only needed for phase 2 trials, which is the case here. We're talking about phase 2-like designs. For some reason, they say that confirmation is not needed for phase 3 because possibly because of randomization. But that's let's say a parallel consideration whether this is appropriate or not. But the answer is yes. We consider confirmed CRs and PRs.
All right, excellent. Thank you very much for that answer there. Another question here asking, do these same, in regards to the non-randomized cohort, do these same principles apply when designing expansion cohorts with a targeted drug that has a predictive biomarker?
Yes. I guess it's the same idea. The point is that, I mean, how do you know it's predictive? Because, in fact, thinking about the correct concept of predictivity, this implies treating patients with your drug and some alternative, but also biomarker positive and negative drug patients. So in principle, everything applies, but you have to consider that at this point, you may not know that the biomarker is predictive. So that's what I would answer to this question.
Great, excellent. Oh, pardon me.
I think Else wants to add something.
I think it might just add another layer of complexity or difficulty, that for a predictive biomarker, we want, of course, to have the randomization for all the reasons we talked about, but then we would also like to see the biomarker negative patients. So I think, yeah, like Everardo said, this adds to the complexity, but in essence, it's the same. It applies to the predictive biomarker trials as well.
Great, excellent. Thank you to both speakers for that answer. Another question here. This audience viewer asking, 1019 leads to 95% CI of 0.29, 0.75. So it is still compatible with ORR at 40%. Thus, in such case, we might not be sure how to interpret the internal calibrator. Would it not be useful to consider a formal rule and or sample size to accept or reject the internal calibration arm?
Yeah, well, it's a good question. It must come from a statistician, I suppose. Well, first one comment is that, of course, here in these designs, we set the type 1 error very liberal, to 10% one-sided. So this is a 95% CI. What I would like to see is the one-sided 90% confidence interval, but this is the first technical answer. But indeed, this intermediate solution is what it is. It does not give us as much confidence, of course, as the fully randomized trial, and in some cases we will still be in doubt. It can just help in some situations to help us decide, but as it's not a formal comparison, it's still only an informal guide to further development.
All right, excellent. Thank you very much for that informative answer, Els. Another question for our speakers here. This viewer starts off by saying, thank you very much for the informative talk, and then follows by asking, for multiple expansion cohorts, can we be flexible about including randomized part for one tumor type and not for the others?
Yes, I think this is, of course, this is possible. If you conceive the different cohorts as independent trials, then you can take a decision for each trial independently as the sponsor see fit. So yes.
All right, excellent. Thank you. Another question here. How do you decide whether the internal calibrator is consistent with design hypotheses? Maybe by formal testing?
Well, it would be nice if we could do the formal testing, but again, it is an intermediate solution, so there's no power to formally test anything. Of course, I'm in favor of having more power and a higher sample size, and a proper randomized trial, and then include some more analysis to stop the trial for futility if needed. But no, this intermediate solution does not offer the opportunity for formal testing.
All right, excellent. Thank you for clarifying that, Els. Another question for our speakers here. This viewer asking, is there a reference or journal article that we can read that explains the randomized design that uses an internal calibrator?
I'm looking at Els here. We don't think so. But I mean, I think not addressing this directly, I think this kind of reasoning is, let's say, embedded in many papers, one way or another, but we haven't come across one specifically devoted to this, so we might think to write it. Maybe we could write it.
All right, definitely a possibility there, but thank you for clarifying that answer, Everardo. And another question for you and Els here. Going back to the recent question with the non-randomized cohort, what about situations where response rate is not considered a sensitive endpoint?
Well, yeah, I can take that one. I think it's more of a clinical, let's say, endpoint issue. It's a good point. We refer to response rate because it's, let's say, a traditional phase 2 endpoint, but also because it's a binary endpoint, and it when computing sample sizes, one tends to get lower sample sizes than, for example, time to event endpoints. But there would be alternatives. Unfortunately, using time to event endpoints as such, for example, progression-free survival or PFS, is a bit more complicated. On the one hand, because for single-arm studies, you cannot compute the sample size, at least using traditional methods and software. If you have a time to event endpoint, you would have to dichotomize it. For example, instead of talking about PFS as such, you would have to talk about PFS at 6 months. So you transform it into a rate, but then you come back to the binary endpoint. And then if you would compute the sample size for a randomized study, of course, the sample size would be in general larger than when you have a binary endpoint. But there are alternatives still in terms of binary endpoint. There are drugs for which maybe responses are not expected, or many responses are not expected. This is the typical situation of what people sometimes call disease control rate or clinical benefit rate, where you add the proportion of patients also with stable disease. So this is one option for immunotherapy. Another option is the durable response rate. So it's you assessing the duration of response, but not as a time to event endpoint, but rather the rate, the proportion of patients with a minimal duration that you pre-specify. So I guess there are other ways to use binary endpoints, not necessarily the complete plus partial response rate, which is the objective response rate, but but other types of endpoints that may be more relevant to certain drugs.
All right, excellent. Thank you very much for that in-depth answer, Everardo. Another question here. This audience member asking, can you increase the sample size if the randomized results are very promising and try to have a definitive comparison?
Yeah, so I think this question means that, well, my answer is twofold. If you have foreseen sample size, reassessment, and so forth in the protocol, and when it's embedded in the design, then you can do so, because then you have taken all the measures that are necessary for the type 1 error to conserve it properly. Now, on the other hand, so then it would be an adaptive design, and that of course is also possible for an expansion cohort. Now, if it's not foreseen in the design and you decide after the trial or somewhere just to increase the sample size, then I think this is still fine, as long as you, from an analysis perspective, you reset the clock and you only analyze your final results starting from that point onwards.
All right, great, excellent. Thank you, Els, for that informative answer. And thank you to both our speakers for all those answers and a wonderful Q&A session. If your question wasn't attended to during the Q&A session, the team from IDDI will do their best to follow up with you after the webinar. If you have further questions, please direct them to the email address showing on your screen. Also, for any requests regarding slides as well, please feel free to contact dominique.grisard@iddi.com. Thank you, everyone, for participating in today's conference. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen, and your participation is appreciated, as it will help us to improve our webinars. I've just sent everyone a link in their chat box. You can say thanks to our speakers and share this webinar via LinkedIn by clicking on that particular link. Now, if everyone would please join us once more in thanking our speakers, Everardo Sad and Els Colt. Thank you again both for an excellent presentation. We hope you all found today's webinar informative. Have a great day, everyone.